team. Part of it is located in Plön, and part of the group is here in Wilhelmshaven with me. Yeah, so my research is mainly driven by my major interest in avian biology, and in particular, the phenomenon of bird migration. And with my research, I aim to link phenotypic traits to their underlying molecular mechanisms and understand the genetic architecture of behavioral phenomena, basically. So the behavior in focus of my work is migration, and by migration, I mean coordinated, directed, periodic, and mostly seasonal mass movements of all or a fraction of individuals of one species or population. And migration is actually ubiquitous in the animal kingdom, and you find migratory species in almost all taxa. So you see monarch butterflies on this slide, and they master this fantastic migration where one migratory cycle actually takes several generations to be completed. Fish do migrate, bats, turtles, and also birds. So it's hugely variable across the animal kingdom, but also within one taxon. And here, for example, birds, migration behavior ranges from these spectacular mass migrations of flocks of starlings that you can see here, and they migrate during the day, and this is what we usually witness. But much of bird migration actually takes place at night and invisible to us. So this is what fascinates me most. So most passerine migrants actually migrate at night, and they do this alone. And particularly fascinating are young songbirds on their first migration. So these birds travel at night completely on their own without any guidance of parents or conspecifics, and they are heading to an area that is thousands of kilometers away and very often on a different continent. And they have never been to this area before. Nevertheless, they really do this with amazing precision. And I keep being amazed by the question, how do they do this? Like, how do migratory birds know where to go? And how do they do this? I mean, their performance is much better than ours was before the emergence of GPS satellite technology. And that's basically the central question that motivates our research. So we know that birds possess a couple of adaptations that allow them to cope with the challenge of migration. So for example, they have at least an inherited initial migratory direction, and we know from behavioral experiments that migratory birds can use many different cues to find their way on this journey. So they use celestial cues like the sun or the stars, and other external references such as, for example, the Earth's magnetic field that is ubiquitous. And in addition to this inherited initial migratory direction, birds also possess an inherited time schedule. And timing here has actually two components. So it determines when to start the migratory journey, but also when to end this journey. And by this, timing also encodes distance. So the birds basically combine this information into a spatiotemporal orientation program, which tells the bird when to start, in which direction to migrate to, and when to stop. So most of our work actually focuses on European blackcaps. That's a very common breeder in our latitudes. It can be kept and assessed in cages. So the male has this characteristic and name-giving black cap, and they basically have a beautiful song, and the female's crest is maroon. So they're very common. You've probably seen them around, but we don't study them because they are only like very cute and nice, but they are particularly interesting for us because within the same species, you find populations that basically exhibit the entire range of possible migratory phenotypes. And if you want to understand migration on the behavioral level, then this phenotypic variation is of your focal study species is basically a key element. So I depict this here in the sketch. So Blackcaps show exclusively migratory behavior in the north of the distribution range, and then the further down south you go, then they become medium distance migrants, short distance migrants. We also have partial migratory populations and also resident populations like the one sitting here in Gibraltar, and there's also island systems that do have sedentary blackcaps, and it's all the same species, and you can really not distinguish them. And in addition to this variability in the propensity to migrate and the migratory distance, blackcaps also show differences in their migratory orientation, and they exhibit at least one so-called migratory divide. And a migratory divide is an area where neighboring migratory populations of the same species that have clearly different migratory programs, and they meet, mate, and produce offspring of mixed ancestry. And populations of blackcaps that are breeding to the west of this migratory divide basically divert towards the southwest in autumn, and migratory populations of the blackcap that breed to the east of this migratory divide divert into a southeasterly direction in autumn and try to avoid the Mediterranean, basically. And since very recently, blackcaps have also shown a very interesting new phenotype. So you find increasing numbers of blackcaps that instead of flying south in autumn, they actually go northwest. So they migrate to the British Isles and overwinter in the UK. And this phenotype is really something that we don't understand very well at all. It's super fascinating to study because it seems to be evolution that is ongoing, and you can really study evolution in action. So that's pretty cool. And the difference in migratory strategy in blackcaps presumably reflects some differences in direction and strengths of selection. And so they're excellent study systems for us to trying to understand the genetic basis of migration, because these traits like propensity, distance, and orientation are extremely variable, and they are heritable. And we know that they are heritable by a large-scale series of crossbreeding and selection experiments that have been carried out in the 80s and 90s by Peter Berthold and Andreas Helbig mainly. And they basically took or brought birds in from populations that show a southeastern and southwestern autumn migratory orientation. And they kept these birds in cages and reared offspring with selectively bred pairs. So the offspring was reared in captivity and isolated from their parents, so to exclude basically the component that they could learn anything from their parents. So anything these birds, these first generation offspring birds know, is what they inherited from their parents. So depicted here are so-called orientation diagrams. So it's a circular plot, and each dot resembles the main orientation of one bird, and the mean orientation of the population is shown by these arrowheads. And you see that the orientation preference of the first generation offspring basically resembles that of the parental generation. And these results clearly demonstrate that there is a genetic basis of migratory direction. And then obviously they were also interested in what's happening when you crossbreed birds from either side of the divide. So what's the offspring doing then? And this is exactly what they also did. So they reared crossbred offspring in captivity, isolated from their parents again, and they could show that these birds show an intermediate phenotype. And if you want to understand the genetic basis of migration, it's, well, can be quite an overwhelming thought because migration is quite a complex phenotype. And in order to nail down the genes, you want to have a rough idea of how many genes you are after in the end. And here I plot the phenotype distribution of the second generation offspring, and it basically picks up on the parental orientation preferences and also still keeps the intermediate phenotype. So this is indicative for a gene or like for a trait that is governed by only few genes with large effects. So this seems to be manageable. And that's basically what we want to understand. So we want to understand the genes behind this. So what is this that tells a bird to go in this direction or in the other direction? And one limitation of previous studies is that the phenotype of these migrants is mostly identified using indirect methods, such as these orientation funnels that I just showed you some results from, or stable isotope signatures, or ringing recoveries where you have only the location where the bird was ringed and recovered. But to characterize the underlying molecular machinery that modulates this variation in migratory behavior, you want to really quantify timing, orientation, and all these traits that you want to characterize as accurate as possible, and do this in the wild. And here we were heading out using light level geolocator technology, that's archival tags that record light intensity. And we basically wanted to characterize variation in migratory behavior in the wild, and then use this phenotypic characterization to map it to the genome and to basically use this variation to understand how orientation and timing is regulated. So I said, this is geolocator technology, and I just give you a bit of an info what I mean by that. So geolocators are archival tags, and they have been miniaturized for the use on small songbirds, such as the blackcap. They weigh less than 0.5 gram. And they basically record light intensity. So here you see the light stalk that sticks out of the feathers, and that basically records accurate light intensity fixed to exact date and time. And then you can convert these values into day lengths and the time of midday, the azimuth. And then day lengths can be used to estimate latitude, and time of midday can be used to estimate longitude. Longitude generally is more accurate. And then by this time series of light intensity data, you can decode or trace back where this bird has been migrating. But in order to get these data, we need to catch the bird back and download the data, because the data is stored on that device, on that data chip, on the bird. So it doesn't help if you see this bird flying around, you really have to have it in your hand in order to download the data. And that's quite labor intense. So I'm just saying this because we can't use GPS satellite technology or so, because there the tags are usually still around 5 grams or so, and our birds weigh 15 grams. So if we would put these devices on these birds, they would never end up in the wintering areas. So the study design we were following with this technology is, it's a highly international and highly collaborative approach. And the study is running since 2016. Until now, we actually have successfully recaptured and traced back the tracks of more than 100 blackcaps. And for some of them, we have followed them over consecutive years to assess repeatability. And the focus of this effort is twofold. One focus, we wanted to characterize orientation across the migratory divide and see what's going on in this hybrid zone, in this area where these neighboring populations that follow different routes meet. And then in a second focus, we wanted to characterize these northwestern migrants and get a better understanding of what this phenotype is doing and why birds are doing this. So here I give you an overview of what we found in the divide area. Plotted here is a subset, basically representing all possible directions from the recaptured bird that we tracked from the area of the migratory divide. So these are not the actual tracks because it's going to be too messy. It's basically connecting breeding and wintering latitude that are indicated with these dots. And these birds contain birds that show the pure southeast and the southwest grounds. But they also highlight that there is a lot of intermediate routes present in the area of the divide, basically confirming what has been suggested in these crossbreeding experiments that have only been done in funnels. So this is really happening in the wild. And it's also, for us, it was like very revealing, very relieving also when we had our first bird that was following an intermediate track back in the hand, because some people have warned me, said like, well, this is probably sacrificing survival because there is ecological barriers these birds have to cross. And there's probably a selection against this phenotype, but luckily, so survival doesn't seem to be compromised, because if it were, then we wouldn't have these tracks, because as you remember, geolocator tracks only can give you data for birds that actually successfully return. Otherwise, we will never get hand on these data. So we are now using this variability in phenotype to map the phenotype data to the genotype, or like to the genome. Basically, we analyze 10x genomic sequencing data, and we'll use these phenotypes to narrow down on seeing where in the genome is orientation regulated in the species. So when we were tracking blackcaps from the area of the migratory divide, we were lucky enough to actually have one bird that was showing this funny phenotype of migrating to the British Isles to spend the winter there. But this was one of very many and not very efficient. And so we wanted to see how prominent is this phenotype across the breeding distribution of this bird, and where do they actually breed? Because this was completely unknown. So it's basically based on some anecdotal evidence and very few ringing recovery data. And it was assumed that they mostly come from this area of the hybrid zone. So which basically our recapture here confirms. But in order to get more data, we decided to go to where they actually overwinter, because this is what we know. So we know they overwinter in the UK. Usually you try to follow birds from their breeding distribution or from their breeding area, because they are more reliable to come back to the same area. And for winter, we weren't totally sure on how this is going, but yeah, no risk, no fun. So we headed out. And the number of blackcaps spending the winter in the UK instead of anywhere in Southern Europe or Northern Africa is increasing. So that is something that was observed, and that is just a fact. And the wintering birds there in the UK show a strong affinity for gardens, especially those with bird tables. And I mean, of those you find many in the UK. You really have like feeder paradise. So why do blackcaps do this? So why do they migrate north in winter? And where do they actually come from? So we went into these gardens and fitted blackcaps that were feeding on these feeders, but also on other food sources with devices. And then we're hoping for them to return in the subsequent winter. And that's also running since the winter in 2016-17. And by now we have 33 tracks of 30 blackcaps. So here we also have birds that we could follow over subsequent winters or subsequent years, basically. We also equipped a control cohort with colorings to see if there is an effect in return probability from wearing a geolocator, but we basically had birds happily returning in equal frequency, so there was no effect of wearing a geolocator, which is, of course, great. So where do they breed? So here you see the dark black lines, and they show you that the overwintering birds that spend the winters in Britain basically breed all over Europe. Yeah, so there is this huge variability of breeding grounds. And that was absolutely stunning to us, and it was totally unexpected. So, well, we assumed, okay, they come from one population somewhere in the area of the divide, but that is by far not true. So not only do these data clearly confirm the existence of this migratory phenotype in actually decent numbers, but what was really surprising was to find this enormous width of the breeding distribution of these UK overwinterers. So there is this one bird which was breeding close to Warsaw. We have birds breeding in Italy. And then they return to winter in the UK. So it's really funny why they do this. But they apparently do this and do this successfully. So we now can use this amazing data set of phenotyping data of blackcaps that we tracked in the wild to compare autumn versus spring behavior. And in the future, once devices maybe become a little smaller, we can also see how the track of a juvenile compares to when it gains experience and ages. Then we can maybe also track families. That would be super cool to do. And also ideally include non-returners to see where selective disappearance is happening or so. And then we can use these data and map it to their genome. So we obviously take blood samples from all these birds to sequence their genomes. So what I've shown you so far is how we characterize a phenotype in the wild. And to match this phenotype to genotype, we have laid the groundwork by heading out to assemble a reference genome for the blackcap first. And so we have an assembled and annotated high quality reference genome that is by now characterized down to the chromosome level. It's really it's a really nice reference, and it forms the backbone of all the analysis that we do in order to understand the genetics of migration. And in a first set of analysis, we obtained whole genome resequencing data from over 100 blackcaps spanning the full spectrum of migratory behavior. So you see these colorful dots, they indicate different phenotypes, like different lengths of migratory journey and different orientation. And we also wanted to place the blackcap in a phylogenetic context and identify origin of variation in migratory behavior and the demographic history. And we also included additional species, so the garden warbler and the hill warbler. They are the closest sister taxa of the blackcap. And we also included the willow warbler as an outgroup. And we used this reference genome and the whole genome resequencing data to characterize population structure, evolutionary history, demography, and genomics of migratory traits. So we want to understand what's driving this variation. And to visualize population structure and relationship, I just show you PCA here, and the colored dots are coded with respect to their migratory phenotype. And resident island populations you see in red, the resident continental populations you see in yellow, and then you see this colorful bubble of all sorts of migrants. And results from this analysis suggest that the difference within blackcaps is mostly prominent in the comparison between migrants and residents in both the Atlantic islands and further south on the continent, like this population in Gibraltar that I indicated when I showed you those birds on the map. And specifically, PC1 does separate resident islands from continental populations and does so like island by island. And PC2 seems to split resident continental birds from migrants, and migrants themselves are not clearly distinguished on either PC here. So there seems to be something in the genome that makes a migrant. So we want to understand the genetic architecture of migratory traits to identify specific genomic regions that have signatures of selection that stand out on this background of generally low differentiation. And here's something funny, I think. Never mind. So overall differentiation is low and limited to the comparison of residents, basically. So we only find 9 outlier loci, and 6 of them seem to be adaptation to residency. And we use some sort of tree-based method that detects differences in haplotype frequencies. So every of these colored lines is basically a haplotype. And you see that there is many of them, so it's very colorful. And in the population that is under selection, or where one haplotype is dominating, you see an increased branch length here, for example, in the resident continents. And this yellow haplotype is dominating here, but you also see that this yellow is also occurring in the other population. So there isn't anything fixed for one population, and this is confirmed by like classical FST-based population genomic approaches. So this is PBS, that is basically an FST-based statistical test that can test more than 2 populations. And concerning the actual identity of genes with regions under selection, several do have logical functions related to the transition from migration to residency. And I picked up on that a bit. So it's really important to note that these variants are not fixed, and selection seems to make use of standing genetic variation. And this could provide a mechanism by which rapid changes that we do see in blackcaps, such as, for example, adaptation to out of a sudden migrate northwest and anything else that other birds are doing, could be solved. And one example of these loci that stand out from the background of very low differentiation is this one that has a very funny acronym. It's a modulator of neuropeptide Y, and neuropeptide Y is located in the brain of birds and does control energy balance, and it stimulates appetite, food intake, and fat deposition. So it has been hypothesized that the effects of NPY may extend to seasonal changes in energy balances that might also be important to migration, including hyperphagia and fat deposition. So migratory birds prior to the migratory journey really eat a lot, and then they accumulate these fat storages on their belly. And beyond its role in energy balance, neuropeptide Y could also facilitate learning and memory. And it does so by some modulations in hippocampal activity. Also has an effect on circadian rhythm, reproduction, and muscle control. And it has been suggested that a common genetic mechanism or any sort of major regulator may control bird migration, and a protein such as an NPY could fill this role. But this is like very speculative, but it's just something that could work out very well. So far, I've considered basically all migratory traits that we are focusing on in unison together. So we want to understand propensity, orientation, and distance. And our results relate mostly to residents. So an elevated population differentiation between residents and migratory birds could reduce our power to identify selection, specifically in migrants. So in the PCA, we had this like colorful cloud of all sorts of dots from migrants. So accordingly, we restrict our analysis to migrants with varying orientations here. And interestingly, many of the signatures that we depict, that aren't that many, are limited to northwest migrating birds. So 4 out of 6 regions that we identified as divergent were actually under selection in the northwestern phenotype. And these regions also exhibit extreme values in FST statistics. So here similar to what I showed you for the comparison between migrants and residents, I also show you this here for within migrants, branch-based statistic that visualizes haplotype frequency. And here it's the same. So there isn't anything fixed in any of the populations, but it seems to be working based on taking advantage of standing genetic variation. And here it's even fewer genes. So basically the list of genes in genomic regions under selection in this analysis here only within migrants is even smaller, but also includes genes with functions that strongly relate to the phenotypes they are associated with. So for example, one gene is SDC1. It's a region under selection in northwestern migrants. And this gene codes for transmembrane protein that regulates signaling in the wind pathway. And that plays a role in feather and beak morphogenesis and probably also feather mold. And if you recall, these northwest migrants are this funny population. I mean, it's actually not a population, but this funny phenotype that winters in the British Isles, and they have been shown to have rounder wings and more narrow beaks compared to the southwest and southeast migrants. So that could be something that where we already see adaptation on the phenotype. They also probably have a different timing of migration and also mold and other life history traits. So phenotypic variation can actually be achieved by diversifying the expression of each component of this genetic regulatory network. And that can happen on different levels. And so far, the focus of data that I presented here has been on identifying genes or genomic regions that harbor coding variation with relevant consequences for the migratory phenotype. And the basis of migratory phenotype difference might not necessarily exclusively be found in DNA sequence. So few genetic changes can result in large differences in gene expression. And as a key complementary approach, we are using gene expression profiling analysis and also characterization of chromatin modification to investigate to what extent variation and evolution of migratory traits is basically manifested by substantial differences in gene expression, either through slight genetic differences or governed by epigenetic processes. And brain is a key organ that processes and integrates information relevant for bird migration. And consequently, our initial focus is on characterizing gene expression profiles in focal brain structures that are assumed to regulate migratory behavior. And specifically, we focus on the hippocampus that's involved in spatiotemporal memory, cluster N. That's a forebrain structure that has been shown to be involved in magnetic compass orientation, and the hypothalamus. And specifically in there, the SCN or AIRs that are also involved in controlling food intake and hormonal regulations. And then we contrast these different phenotypes that the blackcap basically exhibits, but also make use of the fact that the migratory phenotype itself is only expressed during certain times of the annual cycle. So in addition to comparing migratory phenotypes or migratory behaviors, we can also contrast these expression patterns at different times of the migratory cycle, so to illustrate this, I plot sort of the activity profile of a normal blackcap, like during the year outside of the migratory season. And then this red line follows the light intensity. So basically hills are days and valleys are nights. And the blue scribble is the activity profile of the bird. So birds are happily feeding and hopping around during the day. They rest during the night and they again start moving during the day and then rest during the night. So that's the normal profile of any sort of blackcap. But once the migratory season approaches, these birds show so-called migratory restlessness behavior. So that means when they would normally go and migrate, a bird or a migratory bird that is kept in a cage starts to exhibit this inherited behavior and really goes crazy at night. So it wings, it whirs with its wings, it hops around in the cage. It really shows extremely elevated activity levels during the night, and it really happens once the migratory season approaches. It coincides very well with the timing of conspecifics in the wild. So we contrast gene expression during migration with a control period when the seasonal behavior is not expressed. And then we excise the focal brain region of the, and our experimental design actually allows us to use the same bird to characterize RNAseg expression data and chromatin characterization, either through RNAseg that allows us to highlight open chromatin regions that are accessible to bind any regulatory elements, or through ChIP-seq, where you can see different modifications in the chromatin. And we will, in the next step, go down to the cell type specific level and see if, so now we are focusing on different brain areas, but each brain area is composed of different cell types. So we want to look at each cell type separately. So we can identify open chromatin regions and cis regulatory elements and correlate their differential activity with the differential expression of those genes that they regulate in any specific tissue that we want to focus on. And then we can do this between migrants and compare it during the time when this bird is exhibiting the focal behavior that is migration and when it's not doing that. So that's a very powerful approach to pinpoint really important candidates for migratory genes or genomic regulatory elements. And our data that we've analyzed so far really show that there is a key regulatory role in circadian rhythm regulation, which is super exciting because it is not totally understood on how this change in behavior from being a diurnal bird most of the year to then going on to being extremely active at night without compromising cognitive abilities in a migrant. So how this is regulated, we really have no idea. So, but these data can tell us a lot on how this behavior is really regulated. So I would like to conclude by basically summarizing what I've shown you on different levels of the work we are doing and illustrate how this all integrates. So phenotypic variation can be achieved by diversifying the expression of different components of this regulatory network. And our integrative approach tries to maximize complementarity of these different approaches. So on the sequence level, the de novo assembled reference genome forms basically the backbone of all we do and opens the possibility to link behavior to genotype. And population genomics analysis have shown that there is extremely little genomic variation generally. And ancestral behavior seems to be migratory. That's something I haven't covered here, but it looks like the original behavior is migratory and then it was lost twice. And the differences between the populations are restricted to very few small genomic regions and mostly related to adaptation to residency and colonization of new habitat. And in terms of phenotyping, previous work is exclusively based on bringing recovery data and phenotype assessment of selectively bred offspring. And we use light level geolocator data to track birds individually and really we try to characterize the entire spread of the behavior that the species basically exhibits in the wild. And as I said, the basis of phenotype differences might not only be in the DNA sequence, and few genetic changes can result in large differences on the expression level. And our experimental design on this trajectory allows us to contrast different phenotypes and also making use of the fact that the migratory phenotype itself is only expressed during certain times of the annual cycle. And thus, in addition to comparing migratory phenotypes, we can contrast these expression patterns at different times of the year. And then we quantify RNA expression levels of focal brain structures that we think are involved in controlling differences or different aspects of migratory phenotype. And we link this to a taxic characterization that allows us to highlight open chromatin regions that might only be accessible during migration or out of migration. So we can really see on how the differential expression is regulated. And results from all these approaches are closely interconnected. And I think we really have to focus on all the different levels in order to really understand on what is driving this migratory phenotype. And by using high throughput technologies and powerful behavioral approaches that we do in the blackcap system, that we do use in the blackcap system. So this holds great promise to identify and functionally validate. So to identify focal genes or candidate pathways or so, but we want to functionally validate those. And in one step, we want to exploit also a larger number of strategically chosen migratory species, so of other species. So we want to see, is what we find to be true in the blackcap also a solution that other migratory species have chosen or have found. So we carry out the groundwork in our focus species on the blackcaps, but here we ultimately want to generalize these findings and assess identified candidates in a cross-species comparative approach that allows us to assess repeatability and also assess if migratory behavior really has a common genetic basis. So that was my assumption when I started. I'm no longer so totally sure if everyone is really using the same solution or if it is something that where the similarity or the general main link is somewhere that we haven't yet understood enough to find this linking element. So that might also very well be true. And ultimately, providing a proof of causality will be imperative. And I think genetically manipulating identified candidate genes and regulatory regions in vivo to assess how genetic disruption affects the migratory physiology and behavior will be challenging, but should be facilitated by increasingly accessibility of these functional genomic tools also for non-model organisms. And if we combine this with diagnostic, physiological, and behavioral assays, these genetically engineered migratory birds will ultimately facilitate basically a systematic dissection of the molecular and cellular mechanisms that underlie bird migration and in particular orientation. So I want to finish by concluding that I think that our work that aims to understand behavioral genomics of migratory traits will not only contribute to gaining deeper insight into the phenomenon of bird migration itself, so this huge variability of migratory phenotypes that we find and evolution that is driving this fascinating variability. But perhaps most importantly, I think information on migratory genes could allow us to forecast how animals will respond to environmental changes, such as changes in migratory routes and the propensity to migrate that we already witness, not only in blackcaps, but also other bird species, other migratory bird species. What we find when we search for the basis of genetics, the basis of migration, that will also have an important consequence on our understanding of sensory biology. So once we understand why a bird is migrating southwest rather than southeast, we can also start to zoom into understanding on how these birds maybe use compass cues differently. And it will also have important consequences for conservation management. So we hope that what we learn in a common species such as the migratory blackcap will be able to feed into conservation management plans in order to protect more threatened migrants. So with this, I want to thank my fantastic team and really wonderful collaborators across the world, funding sources, and you for your attention. Thank you very much, and I'm very happy to take questions.